Hello friends, today we talk about the further in nervous system, that is neurotransmitter. In last lecture we discussed about the synapse. What is synapse, types of synapse, synaptic transmitter, how one neuron gives signal to the next neuron. So now today we talk about the further in neuroanatomy or nervous system, that is neurotransmitter. It is the most important chemical which is responsible for nerve transmission or neural transmission. So we can say neurotransmitter is endogenous chemical. It is a chemical, we know it's a chemical, or it is a basically endogenous. Endogenous means a body make itself. Okay, body make itself. So it is called as a endogenous. Endogenous. And one more word is a exogenous. So we not take a outside, it is a endogenously, body produce itself. So it is a endogenous chemical substance which are responsible for a transmission between two neurons or neurons and effector organ. Okay, so it is called as it is a chemical, or it is a messenger molecule. It is act as a messenger molecule, means they act as a messenger. It's same like a postman. Postman deliver postcard from post office to our home. So it is a molecule. Postman is a messenger. So same like neurotransmitter act as a messenger molecule. Okay, so this is most important, because as per your syllabus, as per your exam purpose, neurotransmitter is also important. But on this basis, your pharmacological study, pharmacological studies in future, and as well as your some clinical studies like actions of medicine, actions of toxins. This is all basis on understanding of neurotransmitter. Neurotransmitter act a specific receptor. So functions of this specific receptor is also important. A drug usually they act on receptor. Either they block the receptor, either they increase the activity of receptor. So for your future study about the not only the nervous system but also your pharmacological study, also your clinical data or clinical study, they mainly based on this understanding, because every neurotransmitter work through a specific receptor. Like a GABA act through the GABAergic receptor. Like a dopamine it's a several types of receptor, D1, D2, D3, 5 types of dopaminic receptor. So we have a clear cut understanding of this different receptor. So then we can understand the what is the exact the mechanism, what is the functions of dopaminic receptor, and what happen if receptor is blocked or either they are activated more. So some drugs can activate the receptor for prolonged time. Some drugs block the receptor for some times. So different types of receptor act as a they are many helpful for your physiological functions as well as your study of pharmacology. So first of all, definitions: it is a endogenous chemical substance which are responsible for a messenger, or it's are responsible for neurotransmission between two neurons. We know previously there is a synapse. This is a presynaptic neuron. This is a postsynaptic neuron. And basically we know there is a gap. Neurons are not touch each other. Instead of that, they can communicate. They can talk with each other. So by whenever action potential reach to the terminal part of axon terminal, it is called as a presynaptic knob. We know there is our axonal knob or axonal terminal buttons, many words for this. So this is a presynaptic neuron, basically this is a postsynaptic neuron. So they are not, and between there is a synaptic cleft, means there is a space, they are not touch with each other. So basically they secrete a special chemical in synaptic cleft, and these chemicals bind with this membrane. This is called as a postsynaptic membrane. here. Okay, so those chemical which is secreted by presynaptic neuron in synaptic cleft is called as a neurotransmitter. This is a neurotransmitter. Basically, neurotransmitter is a very simple substance. They made from amino acids, or there is a some few steps of amino acids and the synthesis. So biosynthesis of neurotransmitter is very simple. Many neurotransmitter, it's a basically amino acids which is present in our body as a essential amino acids like a glutamate. Okay, so this is a very basic basic amino acids. So neurotransmitter is a very simple substance as well as they are most complex substance. But right now, more than 150 chemicals identify as a neurotransmitter. But out of 150, there is a very few. It's study well. Okay, so if we talk about the history of neurotransmitter, it is a very interesting. It's a discovery of neurotransmitter start from in a very early in 20th century. Okay, before that, majority scientists or neurologists thought that two neurons are talk with each other with a electrical gap, means gap junctions. Previously we saw there is a two types of synapse. Suppose this is a presynaptic neuron, postsynaptic neuron. So there is a connection, there is a tubule structure. This is called as a microtubule, this is a connections. So before 20th century, before research of neurotransmitter, majority neurophysicians or scientists thought two neurons are communicating with a chemical or with a electrical junction, means gap junction. This is called as a gap junction. But after discovery of neurotransmitter, which is usually in 1921, a Dr. Otto Louis. After the experiment of Dr. Louis or Otto Louis, they find out some chemical substance which is responsible for transmission. Okay, we know there is another types of synapse like this. Okay, but there is no basically some connections. Instead of that, they are communicating with each other. So this communications between this types between this type of synapse, this is called as a chemical synapse. They find out there is some chemical which secreted by the presynaptic neuron and they act on postsynaptic neuron. So what they do, they take a heart of frog, two heart of frog. Okay, so I take it's a famous experiment. Everyone know this experiment. They take a two heart in a container of normal saline. Okay, there is two difference. It is a normal saline. Okay, this is a container A, container B. One heart, they are in the first heart or in a heart, they remain the vagus nerve impacted. In B heart, they remove the vagus nerve. So they produce the denervification, remove the vagus nerve or any neural connection. They remove any neural connection. But in a heart, they remain the vagus nerve as it is. We know a heart is it's oven. There is a SA node which produces the impulse spontaneously without any neurological event. It is produced the cardiac impulse spontaneously. So they remain for sometimes. If we take we take out the heart from inside the frog or any persons. Okay, so due to that reasons we can make a cardiac transplant within few hours. If we implant the heart from another person's, from one person's, within a few time, within a few hours, we can make it. So they take two heart from frog, A heart and B heart. In A heart, what they do, they remain the vagus nerve as it is. In B heart, they remove the all nerve connection, vagus nerve. So then what they do, they electrically stimulated the vagus nerve. They make some stimulations. So after stimulations, the vagus nerve produce some. After some times, they shows the A heart become, heart rate of A heart become decrease. Immediately after, what they do, they're taking some fluid around the heart. So this is a fluid field totally. Okay. So they immediately taking some solutions around the heart A and they put into the heart B. Okay. They adding the A solution into the B solutions. So after what happened, sometimes this heart also showing the heart rate slower. Heart rate also decrease in a B chamber. So what they indicate? What is the conclusions from this experiment? That so whenever there is a vagus nerve are stimulated, they produce some chemicals on heart tissue, myocardium, and they lower down the heart rate. Okay, so it means vagus nerve produce some chemicals. So those chemicals which produced by vagus nerve, they diffuse surroundingly, and they take the surrounding fluid and they put on B heart, and they shows there is also slowing of heart in B. So that indicate vagus nerve produce some chemical, and that time they give a name vagus stroke. Means vagus synthesis or vagus product. This is a German name. It is a basically German pharmacologist, Dr. Otto Louis. So after that time, after further research, they find out they secretes the chemical. It's called as a ACH, acetylcholine. So acetylcholine is the first neurotransmitter which is invented, and Dr. Otto is credited with research of acetylcholine. Okay, and after that they got a Nobel Prize for this work in physiology and medicine. Okay, so this is experiment shows vagus nerve produce some chemical which lower the heart rate, and this same chemicals when we put in second heart, they also shows the same effect. So by this way they find out if there is no connections. Instead of that, this a presynaptic neuron secretes some chemicals, and the chemicals act on postsynaptic neuron or effector organ, and they produce some effect. So by this way neurotransmitter find out or they are discovered. So after that there is many types of chemical which is discovered. Nowadays we have more than 150 types of neurotransmitter or chemical which is act as a neurotransmitter. But out of that very few are understand very well. Okay, so what is the criteria for identifications of neurotransmitter? Identification. So there is a given some criteria. Okay, if someone find out a chemical which is act as a neurotransmitter, we cannot accept it is a neurotransmitter. After they fulfill these four criteria, then and then only we call as it is a neurotransmitter. So first criteria for neurotransmitter is it must be synthesized in neuron or axon terminal, or if suppose it is not synthesis but it should be remain there, it should be there. Okay, we know there is a axon terminal, there is a vesicles, these vesicles filled with the chemical, this is called as a neurotransmitter. So it must be synthesized from axon terminal or it should be remain there. Okay. Second criteria, whenever nerve is stimulated, okay, whenever this nerve is stimulated, it should be secreted from vesicles. Okay, it should be secreted and they act on target protein. This is a postsynaptic neuron. So it should be secreted when active, secreted on activation, and act on target tissue or target neuron, whatever, target either gland, muscle, tissue. Okay. Third, this is acted whenever they activated the secretes and they act on target tissue, but it is similar like that, it is the same happen when it should be given externally. So whenever we inject the, suppose a neurotransmitter is they are secreted by this nerve. Okay, nerve 1, it is a presynaptic, act on postsynaptic nerve 2 or neuron 2. So these A types of neurotransmitter, when they acted, they produce some effect on postsynaptic neuron. But whenever we in experiment we give a same neurotransmitter A exogenously, so they must be produce the same effect which is previously noted here. Okay. So first is it must be synthesized in axon terminal or they are remain there. Second, whenever in activations of this neuron, they should be secret and they act on target tissue. Third, what? Its same actions noted when we give externally for experiment. And fourth is it should be wash out. It should be wash out or it should be diffuse or it should be wash out after the actions completed. So whenever neurotransmitter secreted, they act on postsynaptic neuron, they produce some effect, but after some times, after some times within a millisecond, it should be wash out. They are not remain for permanent here. So then and then only we can say it is a neurotransmitter. So what is the criteria? It must be synthesized, it must be stored, it must be secreted from here and act on target tissue. Secondly, its same actions noted when we give externally for experiment purpose. Suppose we give a norepinephrine on cardiac tissue. Normally norepinephrine increase the heart rate, but whenever we give externally norepinephrine to the cardiac tissue, it must be increase the heart rate whenever we do the experiment. Okay, so this is the third point, and it should be wash out after some time. It is not remain permanent for a year. Okay, so this is the criteria for identifications of neurotransmitter. So how neurotransmitter work, and after that we will see what is which type of neurotransmitter or classifications of neurotransmitter. So we know there is in synapse, we also generally this is the presynaptic neuron, this is a postsynaptic neuron, and here the synaptic cleft. And also we know there is in here there is a mitochondria which give the energy, ATP. There is a vesicles. Previously thought that one axon terminal or axon knobs contain the only one types of neurotransmitter, but recent study shows one terminal shows the more than one types of neurotransmitter, different types of neurotransmitter in one axon. But this study also done a majority same types of neurotransmitter remain in one axons terminal. Like if there is a two neurotransmitter, it may be inhibitory both. or either maybe excitatory. Inhibitory and excitatory not remain generally in a same axon terminal, but it may be happen because this is a not clear idea. Because we cannot study the neurotransmitter because they are stored in axon terminal, not in a blood, so we cannot take up from blood and we can study. Okay, for study of neurotransmitter, it's called as a immunocytochemical study. By this way, we can find out where in CNS where is specific types of neurotransmitter remain more. Okay, suppose dopamine is more in basal ganglia. So this is study, we localize the neurotransmitter in CNS, or where is the more neurotransmitter remain. This is called as a immunocytochemical assay. Okay, so we know there is a vesicles. Also we know they contain the special types of ion channel, voltage operated calcium ion channel. Okay, we know. So whenever now impulse coming from up to downward action potential, what they do, they stimulate the voltage operated calcium ion channel. Okay, this voltage calcium ion channels now activated and the influx of calcium occur, and whenever this influx of calcium occur, these vesicles attach here, they are attached, and finally they secrete the chemical in synaptic cleft. This is called as a neurotransmitter. Okay, and this mitochondria give energy. Okay, and whenever this mitochondria, whenever this neurotransmitter secretes, they bind with the specific receptor. We previously talked, either it is a ion channel or either it is a G protein coupled receptor, because this receptor work through the specific second messenger. This is a G protein. So they either they act through the ligand operated ion channel or either they operated with the G coupled receptor. Okay, if they operated through the ligand ion channel, this is called as ionotropic action. Ionotropic action. Okay, and if they are work through the receptor which is G coupled protein, this is called as a metabotropic action. Okay, neurotransmitter, either they are ionotropic or metabotropic or both. Majority neurotransmitter are they acted both. Okay, they either operated through the ion channel or either they operated through the metabotropic receptor. We will see one by one, but whenever they bind, they produce some actions in postsynaptic neuron. Either they produce the excitatory postsynaptic impulse EPSP, or either they produce the inhibitory IPSP. If they produce the excitatory postsynaptic potential and they reach to the threshold value, they can generate the nerve impulse in the next neuron, or either they produce the inhibitory postsynaptic potential, means they inhibited postsynaptic neuron. Okay, we know one postsynaptic neuron getting a multiple neurotransmitter from multiple axon terminals. So what they do, they summarize they produce the summations. Okay, if I draw here one neuron, okay, this is a postsynaptic neuron. They receive the impulse from multiple neurons. They make a synapse with the multiple neurons, multiple axon terminals. All terminals produce a different types of neurotransmitter. Or all axon terminal produce either the EPSP means excitatory postsynaptic potential, either they produce the IPSP. So what they do, if there is a EPSP is more, so this postsynaptic neuron generate the nerve impulse. And if it is a IPSP is more, or whatever, if suppose they receive the message from 100 presynaptic neuron, out of 100, 80% is excitatory. So postsynaptic neuron generate the excitatory postsynaptic potential. And if it is 20% is inhibitory, means they are overlapped by excitatory. And if it is a majority is inhibitory, so postsynaptic produce the inhibitory postsynaptic potential. Okay, so they summarize, summation, they produce the summation analysis, what is more. After that they produce the effect. So this is about the actions of neurotransmitter. In next lecture we talk about the different types of neurotransmitter, and we discuss about the some detail in all neurotransmitter. Okay, have a break.